Okay, guys, on to our third, fourth, and fifth category in our antibiotics. We've done cell wall protein synthesis. Now we're going to nucleic acid synthesis. And this applies to both DNA and RNA synthesis. All right, some of the drugs within this category actually affect one more than the other. But we'll start with those that are starting with RIF in their names, and it's kind of handy because the R will help us to remember that these inhibit RNA synthesis. Okay, so the RIFs, rifampin, rifamycin, rifampicin, right? These are all going to inhibit specifically the RNA synthesis. Now, it's true that RNA ultimately leads to a protein, but that's not how we categorize these particular drugs. They're in a nucleic acid synthesis category, okay? These typically are broad spectrum, used mainly for tuberculosis, and a lot of times these can be paired up with other drugs. And remember, there are lots of reasons you might actually apply a combination therapy. Here we might use one of these with ethambutol or something like that is a cell wall antibiotic. So it's okay to do that, and it will help. It must have some benefit, right, to apply both of them. And these tend to be bactericidal, right? They kill the cells. All right, so we have our RIFs and RNA synthesis. The R's go together. And we also have other drugs within this nucleic acid group that are known as quinolones or fluoroquinolones, okay? Now the ones that, if I had to pick, and I'm not as familiar as maybe some of you are, if you're out in the hospitals and you're working, then you'll hear of these antibiotics more often. But I have, and I think most people have heard of Cipro before, okay? So ciprofloxacin is an example within the fluoroquinolone group, and there's other floxacins you notice here. But these are going to affect more so DNA, but we sometimes will see an effect on RNA synthesis as well. So I'm not saying that there's lots of subgroups here, but you could, I guess, break them down into RNA and DNA, but the fact is that some of these particular drugs do inhibit both, okay? So there's a little bit of gray area here between, saying that they're DNA only or RNA. So I'm not going to expect you to know that kind of detail. But some of them do both, and they're a broad spectrum and bactericidal, okay? Now, these do show a little bit of overlap with inhibiting the nucleic acid synthesis of the host, okay? It's not as specific sometimes as we might want it to be for the bacteria that we're trying to get rid of. For this reason, there are certain hosts, certain people that may have infections with that bacteria that would not be a good candidate to use these nucleic acid synthesis drugs. And this is because it could get in the way of their own nucleic acid production, their own DNA replication, and so on. So a lot of times you don't want to give these kinds of drugs to children because they are actively growing. Particularly during adolescence, you're going to see a really active spurt of growth. Pregnant women, you wouldn't want to have these kinds of drugs because it's going to inhibit, again, some of that DNA synthesis, that replication that would be taking place in the womb, okay? So there's certain people that are candidates for these kind of drugs and they're effective, and others where these would not be, people would not be a candidate to use that particular drug, okay? All right, we have a fourth category, and that is the disruption of cell membranes, all right? A lot of mixins, this end of the word mixin is going to be cropping up a bit here in this particular category. Now, The mixins are a bit of a narrow spectrum. They affect gram negatives more than they do gram positives. This is based specifically on certain kinds of lipids in the membrane. Okay, this is how they work, and this is how they can be more targeted towards the microbe. Because we all know cell membranes, plasma membranes are around all cells, including my own, right? So this is how it can be more specific for the bacteria, is that these mixins are going to narrow in on certain kinds of fatty acids that are in the lipids of the membranes, specifically of bacteria, like the gram negatives. And of course, if we disrupt cell membranes, we increase the permeability of those membranes. It's basically going to compromise the boundary of a cell, and it is not going to survive that. This is going to kill the cell. All right. I remember using mixins. Mixins can actually affect fungi too, and some molds. We had some solutions in my laboratory back when I was a grad student, and we would always put a little bit of this kind of drug into that bath, that solution, because we all kind of use that bath for the molecular biology techniques we had to do, and we didn't want to have to constantly make new. So we didn't want the mold, so we'd add a little bit of this stuff, and it would keep it for a while longer. Okay. And this doesn't have different subgroups, so all you got to do is know the category and the one example within the category. All right, so I told you it would get easier, right? We had 5 subgroups with cell wall. We had 4 subgroups with protein synthesis. You could kind of argue there might be 2 RNA and DNA synthesis in the third one, but now we have no subgroups at all. Just the category and example is what you need to know. Okay. All right. Our last major group, is the inhibition of metabolic activities. So we want a drug that's going to interfere with a metabolic pathway, and we'd want to be smart about it, right? We wouldn't want to choose to inhibit a metabolic pathway that we're dependent upon. We want to choose others, and one of the examples would be interfering with folic acid synthesis. Now, this did crop up back when we talked about the inhibition of enzymes, competitive, non-competitive inhibition, and this was a competitive inhibition, and it's through the use of sulfa drugs. All right, sulfonamides, this would be one example of a drug in this category. Trimethoprim is another. Now, if you pair those two up, some of you might have heard this before, you get something called Bactrim. Okay, now if I were to ask you an example of an antibiotic that comes from Bacillus species, one answer could be bacitracin, right? A topical ointment, kind of toxic if you bring it into the system, but it's very effective on the surface. But Bactrim, despite the fact that it starts with BAC in the word, Bactrim is actually a combination of 2 sulfa drugs. Okay, so don't let that fool you when you see the BAC in the word. It's actually a combination of sulfonamides and trimethoprim. And Bactrim is pretty commonly used for UTIs, lung infections, ear infections, intestinal problems. It's a broad spectrum, as you might guess, being used for all those different kinds of infections, and it's bacteriostatic. So it pauses the growth of that population. All right, so through the years we have developed lots of ways, obviously, right? 5 whole categories going after all sorts of different things in these bacteria. And actually, later in the 1900s, this is a quote I like to bring up here, is that the time is ripe to close the book on infectious disease. This was a statement by a rather important person in the medical realm, and that was the Surgeon General to the Congress in 1969, year I was born, in fact. We were having huge success through the 1900s. After Fleming's identification of penicillin, we identified all these different kinds of antibiotic classes. And we were having great success going after pretty nasty diseases. Tuberculosis was one of them. They thought, well, by the 70s, it was going to be gone. Well, is it gone? Antibiotics are not saving everyone, right? I mean, you hear this stuff in the news. There's resistance to the antibiotics cropping up. And so this is a rather short-sighted statement, I would have to say. Now, of course, this is hindsight is 2020, right? To look back and see what's happening now. So what we're going to do next, when we come back in the next clip, is we're going to check out what are the ways that cells protect themselves. We now know what kills the bacteria, going after these 5 different aspects of the microbe. But now what I want to do with you guys is explore what are the ways these bacteria are getting around these drugs that we've been developing. All right?